In this video, we're going to look over the polysaccharides now. So a polysaccharide is just multiple monosaccharides linked together by a glycosidic bond. These are also known as glycans or oligosaccharides. And you have two different types of polysaccharides. You have your homopolysaccharides, which are made out of the same monosaccharide. And you have your heteropolysaccharide, which is made out of different monosaccharides. Usually in heteropolysaccharides, you only have two types of saccharides, though. You usually don't have heteropolysaccharides made out of three different monosaccharides. Now, to cut these polysaccharides, we have exoglycosidases and endoglycosidases. And it's similar to our peptidases that we talked about, to cut proteins. Same idea. Exo cuts on the ends, and endo cuts in the middle. Now we're going to begin to look at our polysaccharides by looking at disaccharides. And disaccharides are the simplest polysaccharide you can have. Now when we talk about the linkages between disaccharides, we have a naming scheme. First, if it's an anomeric carbon, we'll say if it's alpha or beta. So here in lactose we have beta 1. Then we say what it's connected to. It's connected to carbon 4 glucose, so it's beta 1 4. Now if you have a free anomeric carbon, so in glucose here our anomeric carbon is not bound, we call that a reducing sugar, because this ring can still open. Glucose can still open and become linear again. However, sucrose, alpha 1 beta 2, here we're using alpha and beta both because we have two anomeric carbons in a bond. These are non-reducing sugars. You do not have an anomeric carbon that is free, so no ring can become linear again. So let's look at some common polysaccharides in nature. And first we're going to start with the most abundant, and that is cellulose. Cellulose is made out of two glucose units that are linked beta 1 4. And it's the primary structural component of plant cells. In fact, half the carbon in the world is in cellulose. And cellulose can be up to 15,000 glucose residues long, and they form these flat ribbons. As shown here, this is one glucose polymer. However, what happens is that you have multiple glucose polymers stack up, and they are forming a hydrogen bond network. So all these blue dots are your hydrogen bonds forming. You also get van der Waals interactions as well, and this really means that this cellulose is very compact and it's very strong. It sticks together very well. This is why it can serve as bark, or the outside layer of plants. This is also why we can't really digest it. It's too compact. We don't have the enzymes to break down cellulose. And the enzyme that will do that is cellulase. Now cellulase is really only found in certain microorganisms, and it's very slow, time-consuming process because this cellulose is very tightly packed. So for organisms that do break down cellulase, let's think about the cow. Cows eat grass, so they get their energy from cellulose. However, they have 4 stomachs to do this, and their bacteria will break it down for them. But it takes a long time. That's why you always see them chewing up the grass to just break down the cellulose. Next, we have chitin. Chitin is beta 1 4 N-acetyl D-glucosamine. So it's like cellulose, but the only really big difference is that at the C2 position, we have an amine here. That's why it's N-acetylglucosamine. We have the amine and the acetyl group. And chitin is the component or the exoskeleton of your crabs, crustaceans, insects, spiders. And this is the second most abundant biomolecule there is, because we have a lot of insects. And again, since it's similar to glucose, it will form these layers. These layers are hard, which makes sense why it is the exoskeleton. They're very compact, hard to break down. Now the next polymer we're going to look at is alpha amylose, which is alpha 1,4 glucose. So we were just talking about back in cellulose, that's beta 1,4. Now in amylose, it's alpha 1,4. So all that's different is the position of one anomeric carbon, and this is the energy storage for plants. So starch is what makes up energy for plants. It's in your chloroplast, and it's made out of alpha amylose and amylopectin. So we said alpha amylose is alpha 1,4. Amylopectin is alpha 1,6. So what happens is that you have this amylopectin that is alpha 1,4, and every once in a while, you'll have this amylose, which is alpha 1,4, and every once in a while you will have amylopectin as well, which has these branch points. So really, the big difference is that if it branches, it's amylopectin. If it doesn't branch, it's amylose. And these structures form these tight ribbons. This tight structure is why it's good at storage. It's very compact, and so it can be stored away in chloroplasts. So let's take a look at starch now. So we talked about the subunits, so let's zoom out a little bit. And here's what we were talking about on the previous slide. So in the blue we have amylose. We don't have any branch points. Well, here's amylopectin, and here we have a branch point. So here's another strand of amylopectin. So everywhere you have a branch, we call that amylopectin, while if you don't have any branches, we call that amylose. And so starch has exactly two reducing ends. As a reducing end from the amylose, and amylopectin, even though it's branched like this, it only has one reducing end. And so when we break down starch, when humans begin to eat or digest starch, because starch is how we get our carbohydrates mainly, the first thing we do is that we start to break down starch in our saliva. So we'll randomly hydrolyze these alpha 1,4 glycosidic bonds. Then as we get into the small intestine, we have other enzymes that will start breaking apart the glycosidic bonds, will start breaking down these branch points. While in plants, what happens is that you'll start breaking off glucoses from these reducing ends. So plants will start chipping off glucoses from the reducing end. So these branch points, once you get there, you have another reducing end to start to break down. And so that's one method you can use to quickly release glucose for the plant to use, energy glucose. Now for humans, the way we store energy, one way other than fat, is glycogen. So that's our storage polysaccharide. So we have glycogen in every cell, but it's really in your skeleton muscle and your liver. And this is similar to the amylopectin we talked about in starch, but it's much more branched, every 8 to 14 residues. And what we use glycogen for, we use it for energy, and the enzyme that will break down our glycogen is glycogen phosphorylase. So what it will do is that it will cut alpha 1,4s from your non-reducing end, and since you have multiple branches, you have multiple non-reducing ends, and so you have a way to easily release a lot of glucose at once. While if you didn't have any branch, you could only cut one glucose at a time from each molecule. Here we can get multiple molecules to glucose. And we also have debranching enzymes. So this will break apart your branches, allowing for more non-reducing ends to be available, which allows for even more glucose to be available. And that's it for this video. I will see you in the next one.